questions. If not, this is what and yeah. Start recording. Share my screen. All right, today is October 19th, and we are covering chapter 4 today. Hopefully we'll finish chapter 4, and if we finish early, we'll even get started on chapter 5. I'm not sure. I think we will finish chapter 4, but I'm not sure how early that will be. And then in the lab, I'm going to discuss the unknown projects today. Any questions about what we're doing? Can somebody confirm that you're hearing me?
Yes, we can hear you.
All right, no questions about what we're doing. I'll minimize that. We were talking about endospores last time, and we had discussed how endospores form in a cell. Remember, endospores are a survival structure. They are not a reproductive structure. And I've got a little video, it's not real good, about showing you endospores forming in a cell. Okay, any questions about that? If not, let's move on. We're now going to talk about eukaryotes and their cell structure. We have on the right side a animal cell, and on the left side a plant cell. And they have a lot of organelles which are the same. It doesn't show a flagellum on the plant cell, but actually some plant cells do have a flagellum. And as you remember, only a few animal cells have flagellum. And flagellum isn't unique to eukaryotes. If a prokaryote moves, it has at least one flagella. But the Golgi apparatus and the nucleus are in both plants and animal cells. There are a few organelles which are mainly found in animal cells, like the lysosome, the centrosome. Let me see what else there is. I can't remember off the top of my head what you find in animal cells and what you find in plant cells. I do remember that in plant cells, they have a central vacuole. It's not labeled that here, but this is large enough that that has to be a central vacuole. Central vacuoles are only found in plant cells or plant-like protists. You can find a vacuole in an animal cell. However, vacuoles in animal cells are much less common than vacuoles in plant cells. And that's because animals can pee and release the nitrogenous waste. And plant cells, they just store that waste in their vacuole or in their central vacuole. The central vacuole is the largest organelle in a plant cell. It's larger than the nucleus. Oh, and then chloroplast. How did I forget that one? That's an organelle only found in plant cells or the photosynthetic protists. It's not found in animal cells. Or we're not going to talk much about fungi, but it's not found in fungi either. Any questions about any of that? All right, let's move on. This is an actual electron microscopic image of an animal cell over here and a plant cell over here. And it's really showing you pretty much the same thing. Now, there are mitochondria in both the animal cells and the plant cell. And there's a mitochondria here and a mitochondria there. This table is showing you some of the differences between prokaryotic and eukaryotic cells. Let me blow this up. Wrong one. So we can see it better. Prokaryotic cells are smaller than a eukaryotic cell, generally about a magnitude of 10 times smaller. And typically they don't get down to 0.2 micrometers. Typically they only get down, the smallest is 0.4. There are a few cells which are 0.02, but those are exceptional cells which they think are non-viable when they're that small. And it's a small, I think it's a parasitic protozoa. And when the cells divide, sometimes they're really, really small and they do get this small size, but they think those real small ones are non-viable. And I don't know why the average only goes up to 2 micrometers here, but that's about the average size, 2 micrometers, 1 to 2. And typically prokaryotic cells are much larger, 10 to 100 micrometers. As you know, the chicken egg is much larger, the yolk. There's no nucleus in a prokaryotic cell. There is a true nucleus in a eukaryotic cell. There are no membrane-bound organelles in a prokaryotic cell. They are present in eukaryotic cells. Flagella is in both prokaryotic and eukaryotic cells. The flagella is a little more complex in the eukaryotic cell. They both have a glycocalyx, and for prokaryotic cells, it's a capsule or slime layer. The glycocalyx is present in some cells that lack a cell wall in eukaryotes. So there's many eukaryotic cells that do not have a glycocalyx. Of course, I guess that's true of the prokaryotes too. Most of the prokaryotes do not have a glycocalyx. The cell wall is usually present in the prokaryotic cells. And if it is present, it's chemically complex, largely made up of peptidoglycan, but it does have other molecules. The cell wall, when it is present in eukaryotic cells, it's chemically simple. Like for green plants, their cell wall is totally made up of cellulose. Okay. And then of course, animal cells don't have a cell wall. The plasma membrane is simpler in the eukaryotes. It generally lacks sterols, no carbohydrates. The plasma membrane in the eukaryote is a little more complex. It has sterols and carbohydrates. The cytoplasm, this is out of date. This textbook is out of date, and I'm not sure why it's out of date because this has been known for over 10 years now. Prokaryotes do have a cytoskeleton. Excuse me. It is simpler than the eukaryotic cell cytoskeleton, but they do have a cytoskeleton. They do not have cytoplasmic streaming, and eukaryotes have a complex, well-defined cytoskeleton, and they have cytoplasmic streaming. I'll show you a little video of cytoplasmic streaming. The ribosomes of prokaryotes are smaller than eukaryotes. They're of the size 70S. The eukaryotic ribosomes are 80S. And I will say that you can find the 70S ribosome in the mitochondria and the chloroplast of eukaryotes. And we'll talk about that later in today's lesson. The chromosome in a prokaryote is usually singular and circular. For the bacteria, the chromosome lacks histone proteins. For the eukaryote, there are usually multiple chromosomes. They're linear chromosomes, and they're associated with histones, meaning there are histone proteins in the chromosome. And The protein makes up part of the chromosome. A chromosome is both protein and DNA. Cell division, the prokaryotes largely divide by binary fission. That means the cell splits into two. Eukaryotic cells divide by mitosis. Sexual recombination, there's none in prokaryotic cells. In eukaryotic cells, it involves meiosis. Any question about that? All right, so eukaryotes, if they move by either flagella, cilia, or what's the third one called? Drawing a blank here, just a minute. Pseudopods. The flagella are long. There's usually few of them on a cell. Oftentimes there's only one. If there are many of these projections on a cell, then there's cilia, and they're shorter. They typically beat together, and they look more like a hair, whereas the flagellum looks more like a whip. They're both used for locomotion. Now remember, for a prokaryotic cell, if they move, meaning if they swim, they only have a flagellum or flagella. That's all that prokaryotes move by. But eukaryotes can move by three things: the flagella, the cilia, and the pseudopods. The cell wall is present in plants, algae, and fungi. It's present in some of the protists. Of course, algae are a protist. Chemically, it's simple. There's no peptidoglycan in the cell wall. We're only going to talk about the cell wall of plants or the plant-like protists, in which case it's made up of cellulose. Of course, cell walls are not present in animal cells. They do have a plasma membrane, and it may be covered with a glycocalyx. Okay, any question about any of that? Eukaryotes do have a cell membrane, and it's, I don't know, odd or striking that the eukaryote cell membrane is very similar to the cell membrane of bacteria. There are some differences, but it is thought that the eukaryotes, when they were evolving, they simply used whatever worked well, and they borrowed they think they borrowed the cell membrane of bacteria, and that's why eukaryotes have a similar cell membrane to bacteria. It's mainly made up of phospholipid, and we're talking about the cell membrane of bacteria and eukaryotes. It does contain proteins, and there are peripheral proteins and integral proteins, meaning transmembrane proteins. The peripheral protein is a protein which is on one side or the other of the membrane. Membranes or cell membranes do show selective permeability, and material can cross the cell membrane by three ways: simple diffusion, facilitated diffusion, I guess four ways: osmosis, and that's referring only to the transport of water across the membrane, and then active transport. There is no group translocation in eukaryotes. However, eukaryotes do have another way of bringing material inside the cell or releasing material that prokaryotes do not have. And that is, eukaryotes can use vesicular transport, where they have a vesicle either fuse to the cell membrane and then open up and then release the contents to the cell, or the cell can form a membrane around material outside of the cell and then bring it into the cell. Any question about any of that? All right, let's move on. Eukaryotes have two types of vesicular transport. There's endocytosis, that's bringing material inside the cell via a vesicle. And there's exocytosis, the movement of material outside of the cell by a vesicle, which we call a secretory vesicle, because it secretes the contents of the cell outside of the cell. Any question about that? All right, there are two types of endocytosis, and one is phagocytosis, the other is pinocytosis. Does anybody have an older version of the textbook? Okay, I'm not hearing anyone. The older version of the textbook kind of had that wrong, and they didn't call it pinocytosis. They called it something else, which I don't remember what the textbook called it, and I'm not seeing it in my notes. But material can move into the cell by phagocytosis. That is where the cell literally engulfs the particle by pseudopods, and they call that cellular eating. When the cell puts out pseudopods, which surrounds what it's eating, and then brings that into the cell in a vesicle. You can also have pinocytosis, where the membrane binds to something. It could be a signal, and the receptor binds to it, and then it invaginates the cell membrane. So it folds it in. I'm trying to show it with my hands. Oh, there we go. So the cell membrane's like this, and it invaginates in the membrane. And that invagination just gets bigger and bigger until it's brought inside the cell, and the membrane pinches off, and so the cell membrane reforms, and then underneath it, the cell membrane forms around the vesicle. So that this vesicle, the membrane of the vesicle is made by the cell membrane. And that is called cellular drinking. That's pinocytosis. Any question about vesicular transport? Excuse me. I don't know if you heard that, but I burped there. The eukaryotes have cytoplasm. Of course, cytoplasm is found in the prokaryotes too. It's the aqueous substance inside the plasma membrane, but outside of the nucleus. And that's what the cytoplasm is of a eukaryotic cell. Now, in a prokaryotic cell, the cytoplasm is the internal inside of the cell. There is no nucleus. And we're not going to talk about the plasm outside of the cytoplasm, but it actually has a different name. It's called the nucleoplasm inside the nucleus. The cytoplasm is mostly water, and it is the fluid portion of the cytoplasm is called the cytosol. So the cytoplasm has within it the dissolved materials, and that could be proteins, amino acids, glucose, different salts, other things which are dissolved in the liquid of the cytoplasm that we call the cytosol. Also within the cytoplasm is larger things like the ribosome, but the ribosome is so big that it is not dissolved within the cytoplasm. For example, DNA is usually dissolved, well, I guess that's in a prokaryotic cell, is dissolved within the cytoplasm. In the eukaryotic cell, the DNA would be dissolved in the nucleoplasm. But even though it's a very large molecule, it's usually dissolved in the cytoplasm. But in eukaryotes, when the DNA, oh, what's the word I'm trying to say? It curls up and forms the chromosome, coils up and forms the chromosome, then the DNA is no longer dissolved in the cytoplasm. Any question about any of that? If there's no questions, let's move on. Cytoplasmic streaming can happen in a eukaryotic cell, and that's the movement of cytoplasm and the nutrients in the cytoplasm throughout the cells. And hopefully this is going to run. All right, we're watching a video, so I'll leave it soundless until that ends. All right, cytoplasmic streaming. You can see movement around the cell. And what we're actually watching are chloroplasts, which is a fairly large organelle, and that's why it's easily to see under the light microscope. Now here we can actually see part of the central vacuole. Anyways, while the cytoplasm is streaming, moving around, it can move nutrients around in the cell, as well as obviously chloroplasts. Any question about any of that? I'm not sure why I got that message. It says participants can now see your screen. You did see the cytoplasmic streaming, didn't you?
Yes, I did.
Okay, thank you. Eukaryotes have a very extensive cytoskeleton. It's much more extensive than the cytoskeleton of prokaryotes. The cytoskeleton is a structural framework consisting of proteins running throughout a cell, and it makes the backbone of the cell. In eukaryotes, there are three types of protein filaments. There's microfilaments, intermediate filaments, and microtubules. And let me see if I can blow this up. We can't see exactly where the cell is here because it's showing you only the, well, the cytoskeleton. What is this? Is this the, I think this is the microfilaments. But the cell would be basically roughly this shape here. And throughout the cell, you can see that there's a cytoskeleton running throughout it. And it's the backbone of the cell. You can anchor things down in the cell so that they are not moving, even when the cytoplasm is streaming, for example. And I mentioned a long time ago, I don't know how many days ago, but in the past, that you can have proteins anchored in the plasma membrane, and they'll be anchored either to the cytoskeleton or to the external matrix of the cell. And if they're anchored, they will not move in the plasma membrane. All right, here we can see this cell here. And this is not showing you the cell, but we can see where the cell is because of the microtubules in green and the actin filaments in red. And then this is the nucleus. So wherever there's a blue, there's a nucleus. And so we're seeing several cells here, but this is one cell here, and there's probably one cell here. and then one here, one here, one here, and one there. We're only going to talk about this one. The point is, within the cell, there's a very extensive network of proteins that we call the cytoskeleton. Any question about any of that? All right. The ribosomes are the site of protein synthesis. In eukaryotes, the ribosomes are larger at 80S in size. And these ribosomes can either be membrane bound, and they'll be attached to the rough ER or to the nuclear membrane. And it's actually the ribosomes which make the ER look rough. And so the rough ER has ribosomes on it. The smooth ER is smooth because it doesn't have the ribosomes attached to it. Ribosomes can also be free, meaning that they're in the cytoplasm and they can float around in the cytoplasm. Remember, ribosomes are big enough that they're not dissolved in the cytoplasm. Any questions about any of that? I feel like I'm going to sneeze, so hopefully I won't sneeze while I'm lecturing. Hopefully I'll turn my head and you won't get the full force of the sneeze. Eukaryotes also have 70S ribosomes, but these 70S ribosomes are only found in the chloroplast and the mitochondria. And we'll talk a little bit more about that later. When I say the word organelle, I mean a specialized membrane-bound organelle structure. Because eukaryotes have this membrane-bound organelle, eukaryotes can compartmentalize their inside of the cell to engage in different functions. And each organelle is a compartment or sac, and it runs a specific function for the cell. So eukaryotes divide up the functioning of their cell by the different organelles. A prokaryote cannot do that because it doesn't have any organelles. And so everything is happening in the cytoplasm. The different organelles typically have a characteristic shape and they perform a specific function. The function could be cellular growth, maintenance, or reproduction. Let's take a look at some of the organelles in a cell. And I guess before I go further, there is a non-membrane bound organelle. So that would be a structure that is not surrounded by a membrane. And I don't really like the terminology, membrane bound organelle and non-membrane bound organelle. So I usually call the non-membrane bound organelles a cell structure. And then the membrane bound organelle, I just call an organelle. Any question about any of that? Oops, too far. So here we're looking at the different organelles in a cell, and this is listing both the membrane-bound organelles and the non-membrane-bound organelle. Like a microtubule is a non-membrane-bound organelle. The ribosomes, a non-membrane-bound organelle. Cytoplasm, non-membrane-bound organelle. And I would prefer calling these cell structures. The nucleus, the rough, well, the endoplasmic reticulum, the chloroplast, those are membrane-bound organelles. Any question about any of that? We'll be talking about various organelles. The nucleus contains the DNA and the chromosomes of eukaryotic cells. So its function is mainly to protect DNA. It holds the DNA and protects it. It does have a few other functions like the nucleus regulates what comes into the nucleus, but also regulates what leaves the nucleus, like a messenger RNA cannot leave the nucleus until it's finalized. There's the endoplasmic reticulum, and it has two types, the smooth ER and the rough ER. The rough ER is the same as the smooth ER, except it has attached ribosomes to it. The smooth ER has no ribosomes. The Golgi complex, and we'll talk more about that one, modifies and sorts proteins. And these proteins are received from the endoplasmic reticulum. And then it packages the proteins and then sends them elsewhere in the cell. Any question about any of that? A lysosome is a specific vesicle that stores digestive enzymes. And one of those digestive enzymes is lysozyme, which gives the lysosome its name. Anyways, the lysosome is an organelle for digesting contents, like an invading pathogen. The lysosome can digest it. Come on. Vacuoles are found in animal cells, but they have fewer of them than plant cells. And the central vacuole, which is the largest organelle in a plant cell, is only found in plant cells or plant-like protists. Vacuoles are a cavity, and what they do is they store contents. And I guess I should change this. We call it a vesicle if it is transporting the material. Okay, so if it's moving material around in the cell, we call it a vesicle. But once that vesicle gets to where it wants to stay, and it's now just storing the material, it's not moving it to another place in the cell, then we call it a vacuole. So a vesicle moves material from one place in the cell to another. And then once it stops moving, we will call it a vacuole because it's storing the material. So a vesicle and a vacuole are the same thing, or at least they can be the same thing. And a vesicle can turn into a vacuole, because once the vesicle stops moving, by definition, it's a vacuole. And if a vacuole is moving, by definition, it's a vesicle. Any question about any of that? All right, we'll talk more about the mitochondrion and the chloroplast. Let me mention the mitochondrion is the organelle in eukaryotes that undergoes ATP synthesis and cellular respiration. And the mitochondrion is the cellular powerhouse of the cell because it makes ATP. The chloroplast is only found in plant cells or plant-like protists, and it's the site of photosynthesis. So if a plant cell undergoes photosynthesis, it has to have a chloroplast. Any question about that? The perioxisome is a special organelle. It contains enzymes for oxidation, and it can release toxic substances on, well, an invader or an invader to the host or invader to the cell. and then destroy the pathogen by using something like hydrogen peroxide. Any questions about perioxosome? All right, the centrosome is not a membrane-bound organelle. It is a non-membrane-bound organelle. And the centrosome is involved in cell division. It does make up the centriole, which is the mitotic, it forms the mitotic spindle when the cell is undergoing mitosis or meiosis. At least the centriole forms mitotic spindle in animal cells. I should word it that way. Any question about any of these organelles? So all of them are membrane-bound organelles we've discussed, except for the centrosome. It's a non-membrane-bound organelle. The Golgi complex is found in both plant and animal cells. And what it does is it receives packages of proteins that come from the endoplasmic reticulum, or ER. And then the Golgi complex modifies, sorts, and then packages those proteins up. And once they're packaged up, the protein will leave the Golgi complex and move to where it's supposed to be, either inside the cell by using a vesicle to move it from one location, which is the Golgi complex, to another. Or if the cell is going to secrete the contents, it will form a secretory vesicle, and then that vesicle will go and merge with the cell membrane, and then open up the contents of the vesicle for releasing it into the outside of the cell. Let me try and draw that. Wrong place, just a minute. All right, so you can sort of see here the Golgi apparatus on this side. The vesicles, which are shown in green, are arriving from the ER, especially the rough ER, because that makes the proteins. And the vesicle then fuses with the Golgi complex, and then that releases the proteins inside the Golgi complex. There's a picture there. Although that's not it. So that would be one here, right here, fusing to the Golgi complex. You can barely see that. These are actually leaving the Golgi complex. And then the Golgi complex will modify the protein that could be chemically altering them. And then it'll sort the proteins and then package it up in a region of the far side of the Golgi complex. And then that'll form a vesicle, which will then bud off of the Golgi complex. And then this vesicle will move from one location in the cell to another, transporting the material. Any questions about the Golgi complex? So those proteins that are leaving the Golgi complex will leave in a transport vesicle. And it'll be called a vesicle until it's reached where the protein's supposed to be. And if the vesicle stays around and just holds on to the proteins, then we call it a vacuole, so it'll change its name. But it can also release the proteins, in which case the vesicle will disappear, but the proteins will form their function. Any question about the Golgi complex? I've already talked about lysosomes, so I won't go further. The Golgi complex, to me, always reminds me of a stack of pancakes. So here we've got about 4 or 5 pancakes, and then these big blobs there. You can pretend that those are blobs of butter on the pancake. In reality, what they are, the ones shown here, are vesicles being formed in the Golgi complex, and they'll bud off like that one is budded off. And then it'll go moving around as that one is doing, moving the contents of the cell from one location to another. Any question about any of that? Come on. Can't get it to work now. All right, let me manually do it. All right, eukaryotes have an endomembrane system. This is a system set up by the cell to make contents, modify them, and then transport the contents to their final destination. And how this is done is with the endomembrane system, which is various eukaryotic organelles which work together to make, transport, and deliver largely proteins, but anything the cell is making, from their site of being made to their final resting site. Any question about any of that? Let's see if I can get this video going. It'll be discussing the transmembrane system.
People with ADHD save 10 hours studying
This is
with
a
a Chrome
commercial.
extension. But no, by all means, go ahead, skip.
Welcome to Moo Moo Math and Science. In this video, I'd like to talk about the endomembrane system. The endomembrane system consists of organelles that create, modify, and transport proteins and lipids throughout the cell. It consists of the nuclear membrane, rough and smooth ER, the Golgi apparatus, and different types of vesicles. First, ribosomes, which are protein factories of the cell, can be found on the rough ER. These ribosomes create proteins and then transfer these proteins into the ER, where they undergo modifications, such as folding. These modified proteins may be absorbed into the membrane of the ER or packaged into a vesicle that may travel to the Golgi apparatus. The smooth ER may transport lipids to the Golgi apparatus, also using vesicles. A vesicle is a relatively small membrane-bound enclosed sac that stores or transports substances. At the Golgi apparatus, the proteins are sorted, packaged, and tagged so that they can travel to the right place inside or out of the cell. The receiving side of the Golgi apparatus is called the cis face. The opposite side is called the trans face. The transport vesicles that formed at the ER travel to the cis face, fuse with it, and empty their contents into the Golgi apparatus. As the proteins and lipids travel through the Golgi apparatus, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of short chains of sugar molecules. These newly modified proteins and lipids are then tagged. These tags help the proteins arrive at their proper destinations. Finally, the modified and tagged proteins are packaged into vesicles that leave the trans face of the Golgi apparatus. Some of these vesicles deposit their contents into other parts of the cell where they may be used, while others release their contents outside of the cell by diffusing into the cell membrane. So there we go, the endomembrane system. Thanks for watching, and Moo Math uploads a new math and science video every day. Please subscribe.
Okay, any questions about the endomembrane system? No questions? I'm going to shut that down. Where the heck is my, I don't have the cursor here. Just a minute. See if I can, I guess I can shut that down. There we go. I'm not sure why I didn't have the cursor there, but I wanted this so I can click on the PowerPoint. All right. So in a nutshell, this picture is showing you the endomembrane system. It really starts with the endoplasmic reticulum, and it's largely the rough endoplasmic reticulum because that will be the site of protein manufacture. And then the proteins are made here, and then they're put into a vesicle, and then the vesicle fuses to the Golgi apparatus. You know, this is shown backwards. This should be binding on this side, and then this shouldn't be budding from this side. It should be budding from that side. But you get the picture. Anyways, the proteins are delivered to the Golgi apparatus. The proteins are then modified in the Golgi apparatus. And then they're sorted and tagged, and they move to the far end of the Golgi apparatus. And then they leave the Golgi apparatus with another vesicle. And that's to take the protein to its final destination. If the protein is going to be used by the cell, it will go to the cell. Like, for example, it could take proteins to the nucleus. If it is secreted outside the cell, like in a teenager with their raging hormones, the protein will be made and modified in the Golgi apparatus, and then will bud out. And then that vesicle will fuse with the cell membrane. Here it is here. And then release its contents outside the cell. Any question about any of that? So the endomembrane system involves the rough ER, transport vesicles, the Golgi apparatus, and then transport vesicles again. All right, I mentioned before that vacuoles can be found in an animal cell, shown on the right, and then there's more of them in a plant cell, which, oh, there it is there. It's showing you probably the central vacuole. It's this big thing here. Nope, that's the nucleus, it says there. Boy, that doesn't look like the nucleus. That looks like the nucleus. Anyways, the central vacuole is much larger than a vacuole, and the central vacuole is the largest organelle in a plant cell. As I stated, vacuoles store material. That's their function. They also can provide structural support and storage of waste. Am I going to talk about the central vacuole? No, so let me talk about the central vacuole here. The central vacuole is sort of like our refrigerator where we have nutrients, our food, and of course our liquids. But green plants store more than nutrients in their central vacuole. They also store nitrogenous waste products. So you can think of your refrigerator with food and then urine stored inside of it. And that's kind of weird, but that's just the way plants are. They will store both nutrients and waste products in the central vacuole. Any question about any of that? All right, the mitochondria is more than one. Mitochondrion means there's only one. Is the site of cellular respiration. This is where the cell makes most of its ATP. So we can say the mitochondria is the cellular powerhouse of the cells because they make ATP. Here we're looking at a mitochondria, and you'll notice that it has two membranes, an outer membrane, and then an inner membrane, which is highly folded on itself. Now, this is an actual electron microscopic image of a mitochondria. The cristae is, let me see if I can blow that up. There is a space between the inner membrane and the outer membrane. Let me get the mouse there. And then there's a space inside the inner membrane, and that is called the matrix. The cristae is just the folding of the inner membrane. Inside the mitochondria, you can find DNA and small ribosomes, the 70S ribosomes, which that's not shown here. Any question about any of that? This is unusual where an organelle contains DNA and ribosomes, but both the mitochondria and the chloroplast have both DNA and 70S ribosomes. Here we're looking at a chloroplast. It's the green part of the cell and what makes the cell green. And of course, the green comes from the chlorophyll pigment, which is in the protein of the thylakoids of a chloroplast. Any question about any of that? Chloroplasts are the site of photosynthesis. Once again, the chloroplast has two membranes, an outer membrane shown in green and yellow here, and then an inner membrane which is highly folded, much more so than the mitochondria. And I can't get that out of there. I'm gonna get rid of that. Get this out of there. There we go. The inner membrane is highly folded in on itself, and that we call the thylakoids, which is a stack of three of the folded thylakoids. We call a granum. Okay. Any question about that? And here's an actual electron microscopic image of a chloroplast. And you can see that the membrane, the inner membrane, is highly folded on itself, forming the thylakoids. Within the chloroplast, as I mentioned earlier, you can find DNA, it's a small molecule, and then 70S ribosomes. This DNA is not a plasmid. We'll talk a little bit more about that in just a minute. Any question about the chloroplast and the mitochondria? If not, let's end this show and talk a little bit about why do eukaryotic cells have two membrane-bound organelles which contain DNA inside of them and 70S ribosomes. And that is the point of the endosymbiotic theory. Let me start a new one. I probably shouldn't use this. I should probably use paint. Oh, I got paint open. There we go. We got it there. The endosymbiotic theory is a theory to explain why eukaryotic cells have mitochondria and chloroplasts. And remember, chloroplasts are only found in the photosynthetic cells of photosynthetic organisms, meaning the green cells of a green plant or a green photosynthetic protist. And if it isn't green, it can be another color, and we're not going to go into that, but you can have a different pigment than chlorophyll, in which case the photosynthesis will occur with a different color. Like red algae has a red photosynthetic pigment, and that's why we call it red algae instead of green algae, which has chlorophyll as the photosynthetic pigment. All right, so this is on page 275 or 276 of chapter 10. You can read about the endosymbiotic theory. I will now discuss it. The endosymbiotic theory proposes that a long time ago, before eukaryotes were around, meaning before they evolved, there was a proto-eukaryote and there were bacteria. Let me get my paint thing here. And the proto-eukaryote was a big cell. Whoops, that's the wrong one. Was a big cell. And I'm just going to abbreviate that with proto-E for proto-eukaryote. And there were little bacteria cells. We'll just draw a small bacilli, if I can get this back. There we go. And the proto-eukaryote came up to the bacteria and engulfed it. So we'll go engulf. This is where, let's switch to here. Yeah, we'll just say engulfed. But the proto-eukaryote did not consume the bacteria immediately. It kept it around. Let's go back to here. And so now let me erase this. And draw the bacteria inside the proto-eukaryote. So the proto-eukaryote didn't eat the bacteria immediately. And I should state that the bacteria had its own cell membrane, probably had a cell wall in addition. And it had a vesicle because it was brought into the cell by a vesicle. So it had two membranes, the vesicle membrane and then the cell membrane of the bacteria. And for whatever reason, the proto-eukaryote had glucose in the cytoplasm. That I can write right here. Whoops, that's not it, right there. And some of the glucose went from the cytoplasm to the bacteria. And the bacteria ate the glucose. And then the bacteria made ATP out of the glucose. Let me write that over here. The proto-eukaryote could only engage in glycolysis. And have we talked about glycolysis yet? I'm sorry, I teach so many classes, I forget where we are. Glycolysis is the beginning of aerobic respiration, and it can take glucose and make ATP. So the proto-eukaryote, let me go like this, takes glucose and from glycolysis, which I'm not going to write here, makes 2 ATP. See if I can make that bigger. But the bacteria can take glucose and with aerobic respiration make 38 ATP. Oh, I don't have the number in there. Let me see if I can put that number in now. 2 ATP. So the bacteria could make a lot more ATP out of the glucose than the proto-eukaryote. For whatever reason, probably because life was early back then and it just did things which was not necessarily the best for the bacteria, although it actually turned out to be good for the bacteria later on. The bacteria released ATP into the cytoplasm. We don't know how much ATP was released, but it was many more than 2. It may not have been 38, but it may have been, well, it was someplace between 2 and 38, and it was probably on the high side. So something like 20 to 38. And the proto-eukaryote was now really confused. Oops, didn't spell that right. Because it lost one glucose molecule, but gained, we'll just say gained, a large number of ATP. Many more than the proto-eukaryote could make from one molecule of glucose. Any question about any of that? Where the proto-eukaryote said, well, this is a good thing. This bacteria is making me ATP from glucose and is making a lot more ATP than I can make. So I'm going to keep this bacteria around and then it'll make me ATP. And so the proto-eukaryote fed the bacteria glucose, and then the bacteria released ATP into the cytoplasm for the proto-eukaryote. And then the two lived in symbiosis, the proto-eukaryote and the bacteria. Nowadays, the symbiosis, according to the theory, has happened so long, we can't find the bacteria living anymore, any place in the world on its own. It only lives within inside another cell that we call a eukaryote. So over time, I guess I'll try and draw an arrow. Over time, the two, no, that's not it either. Let me get the arrow. Over time, the two became inseparable. And here we call it then the eukaryote. And that is the eukaryote. Let's make that bigger. So we don't find the proto-eukaryote anymore without the bacteria, and we don't find the bacteria anymore outside of the proto-eukaryote or the eukaryotic cell. So the two have been living in symbiosis for so long that they're together now. Any question about any of that? That's the theory of how the eukaryotes obtain their mitochondria, meaning this bacteria went on to evolve into the mitochondria, and the proto-eukaryote went on to evolve into the eukaryote. So I guess I should put right here if I can. Eukaryote plus mitochondria. Of course, that's with more than one bacteria, but you get the picture. Any question about any of that? Let's quickly end this and say that what is the theory for, what is the evidence for this theory? First, you'll note that the mitochondria has two membranes, which could come about from the cell membrane being engulfed in a vesicle that's brought into the cell. Some other evidence for this theory is that the eukaryotes have histone proteins, the bacteria do not, and the mitochondria and the chloroplast do not have histone proteins. But they do have 70S ribosomes, which the bacteria have, and the eukaryotes have 80S ribosomes. The mitochondria and the chloroplast divide by binary fission, which is the way a prokaryotic cell divides. The eukaryotic cell divides by mitosis. Another few things that are different is the first amino acid in protein synthesis is methionine in a eukaryotic cell. It's formomethionine in a prokaryotic cell, and it's formomethionine in a eukaryotic cell. The DNA is also similar between the bacteria and the DNA of mitochondria and chloroplast, totally different than the DNA of a eukaryotic cell. And then the strongest evidence, if we sequence the DNA of mitochondria and chloroplasts, we find that that DNA is similar to the sequence of DNA of the bacteria, and it has no relationship to the DNA of a eukaryotic cell. Any question about any of that? And then lastly, which isn't evidence, I just want to point out, is something that's odd, is animal cells had this happen one time to get the mitochondria, but plant cells and the photosynthetic protists had this happen twice. First, a bacteria was engulfed by the proto-eukaryote to become the mitochondria, and then they engulfed another bacteria which engaged in photosynthesis, and that went on to become the chloroplast. Any questions about the endosymbiotic theory? All right, if there's no questions, I'm going to end it here, and I will see you at 6:30 for the lab.